Hello. This time I'm explaining you what is artificial chromosomes. In fact, in biotechnology, our purpose is to clone the DNA or express the DNA into some translational product. So how it can be possible? So it is possible by use of the cloning vector and the expression vector. And one of the vectors which are very much used to clone the plant DNA into the yeast cells with the help of yeast artificial chromosomes, and it is called as YAC, yeast artificial chromosomes. So let us just talk about what are the some of the features it has. So definitely, besides behind any discovery, it is the dedicated work of scientists. So these are the two scientists, J. Sastock and Andrew Murray, who in 1983 this, they developed this YAC artificial chromosomes, and this paper was published in the Nature in the same year. So our purpose is to clone the DNA, but always there is the limitations. If the size of the DNA that increases, then you'll have to switch over to the some other cloning vectors. Here are the some list of the vectors and the vectors which are transformed into the host cells. They are introduced into the host cells, and these are the size. So, for examples, in M13 phase, it can carry the DNA in between 1 to 4 kilobase, and within the E. coli, it can propagate. Similarly, the plasmid, it can carry 1.5, 1 to 5 kilobase, lambda phos, 5 to 25 kilobase, cosmid, which is a chimeric vectors, it can clone a DNA of 35 to 45 kilobase, P1 phos, that's 70 to 100, then prokaryotic artificial chromosomes, 100 to 300, bacterial artificial chromosomes, more than or equal to 300 kilobase, and definitely the yak, that is, that can clone a DNA of about 200 to 500. And if the strain tends to vary, because the genomic size of the yeast cells that varies from strain to strain, and if it bears the large size of the genomic DNA, then it can be able to clone the DNA of larger size. So, artificial chromosomes are nothing but it is a chromosomes like DNA molecules, which is assembled in vitro from some of the components from the, you can say, yeast, and some of the component from the PBR322 cloning plasmid vectors. And beside this, some other sequences are added in order to ensure its stable maintenance, like chromosomes inside the eukaryotic cells and as like plasmid in the prokaryotic cells. So basically, if we talk about the plasmid, it is the circular, and if we talk about the chromosome, it should be linear, and this plasmid is converted into the linear form. When this plasmid will be there, it can be cloned within the bacterial cells, and if it is linear, it can be cloned into the yeast cells. So some facts about the yeast is that the yeast has got genomic size of 12068 kilobase pair. It is the genomic size, and it was completely sequenced in year 1996. It translates about 5885 proteins encoding genes are there, and 140 RNA genes are there, 40 small nuclear RNA is there, and 275 transfer RNA genes are there. and the number of the haploid chromosomes, that the 16 linear chromosomes are there, and if it is the diploid, then 2n is equal to 32. And in few plasmids are present of size 631 base pairs, because some is still, they also has got the plasmid like the bacteria, and its number may varies from 70 to 200 cells, and the size is about 6318 base pairs DNA. One thing is very important while you deal with the culture of the yeast cells in the medium. So basically, every microorganisms, they just classify into two types. One is called as prototrophs, another one is called the auxotroph. Prototroph is the wild type, and autotroph is mutant type. It is mutant type. It means that wild types can grow very easily in minimal medium. If a petri plate is there and if the minimal medium is present, minimal medium is present, including NaCl, or you can say potato extract, or you can say agar, and then dextrose. This is the medium, and no other additives have been added. So this is called minimal medium. The wild type can grow to form colony, but auxotroph or the mutant type, when it will grow in the minimal medium, it is not able to form the colonies. So prototrophs synthesize all the nutrients which are essential for the growth from the inorganic material, while autotroph, it has lost the ability to synthesize an organic compound required for its growth. So here you can say, the if a microorganism is histidine plus, that's the wild type, it means it is prototrophs. It can grow without histidine in the minimal medium. But histidine minus, it is auxotroph, cannot grow without hist, and hist is essential to initiate the growth of the colony. So, what is yak? So, yak, you already, it is mentioned here that yak is a human engineered human engineered DNA molecules used to clone DNA sequences in the yeast host. Okay, and yaks are plasmid like that. One of the important plasmid is PYAC3. So this is one of the important plasmid. When yaks are in the plasmid form, then it will act as a shuttle vector. That is, it can propagate the DNA into the bacterial cells as well as in the yeast cells. And the development of yaks are based on the logic that eukaryotic linear chromosomes needs to replicate, and also it must be stable inside the yeast cells. So for that purpose, it never has only the replicating origins, but also they must be a centromere and telomeres to sustain like mini chromosomes. If you recall a chromosomes, if you recall a eukaryotic chromosomes, then metaphase chromosomes, that has got the two sister chromatids. These are sister chromatid. So, and the end is called as the telomere, and the central part, that is called a centromere, which just connect the two sister chromatids together. And telomere, these are essentials for the separating the two sister chromatids. So if that telomere is deleted, then these two sister chromatids may fuse together, may fuse together. It cannot be. So telomere has got so many functions. For example, it prevent the attachment of the two sister chromatids. Also, it is essentials to maintain the length of the telomeric length also, and also this telomere prevent the action of the nuclease enzymes, exonuclease enzymes, and these are only possible due to the virtue of that this DNA, it is quadruplex forms where the 4G they forms a Hugnes base pair G G G G. So this is called as the Hugnes base pair as compared to Watson base pair. Watson base pair that happens in the complementary DNA, but this Sugnes base pair it forms in the single stranded, which forms quadruplex. So if it is single stranded, it can fold in like this to form four stranded, one strand, second strand, third strand, fourth strand, and this fourth strand, if G is present, then it can form the Hugnes base pair. So, due to this, the exonuclease activity is prevented. Exonuclease activity is prevented. So, from where it has been originated? So, this plasmid, this yeast artificial chromosomes has been created from the two. One is from the yeast regions, and another is from the plasmid regions. So from yeast, they have got the centromere, they have the telomere, and they have the autonomous replicating sequence. Okay, these three. Besides some selective markers, URA3, TRP1, SUP4. These are the selective markers. So centromeres again it is for the attachment of spindle fibers during cell division. Telomere to preserve the integrity of the chromosomes by virtue of telomerase enzymes, and these telomeric sequences is has got the six base pairs repeat C C C C double A. So these are repeated in 20 times or 70 times, 20 times to 70 times. And this is essential to prevent the exonuclease activity. ARS, it has got the replicon and it is essential for the replications initiation. And col E1, it is the bacterial or IC or the bacterial replications. Beside this, this PEYAC3, that is yak, also pauses URA3. It is essential for the uracil biosynthesis because it synthesizes some enzymes which are responsible for the uracil biosynthesis. Similarly, TRP1 also synthesizes some enzymes for the tryptophan biosynthesis, and sub4 it is also selective markers which is present in the cloning site. Yeah, you can say if it is the sub4, then within this the cloning site or multiple cloning sites are present. So when a DNA is inserted, so when the DNA is inserted, suppose that this is a sub-region, this is sub-region, and this is the DNA inserted in the sub-regions. Sub 4 here is no insert. So how you to in order to differentiate the clone with insert and clone without inserts, so for that very purpose sub 4 genes are required to differentiate transformed and untransformed colony. And again multiple sites are essentials because with the cutting of this very sites creates a gap for the integration of the any DNA insertion of your interest. So here is the construct of the YAC. It is 11.4 kilobase, and you can say it has got the components from the yeast as well as from the bacteria. So from the bacteria, it has got the ori site and the TRP1. And from the east, it has got the ARS, it has sub4, it has URA3, again, and also the centromere and the telomere. Okay, so in order to make it stable inside the E. coli, it should be circular. So for that, the BAM H1 site they are attached at both ends, and these BAM are sticky end. So with the help of T4 ligase, they are made circular. So this is the circular yak. Okay, and the two telomeres, it has been taken from the tetrahymena, which is also functional in yeast cells. So if we want to just create, because this is circular. So if you want to create uh, if you want to create a linear chromosomes, then you will have to digest it with the BAM H1. So you just recall the yak plasmid. So here was the telomere, here was the telomere, and at that very site, at this very site, the BAM was there. So this is BAM H1. This is also BAM H1. These are restriction enzymes. So when it is treated with BAM, so here it is cut, and after cutting this portion is excised, and we can get a linear chromosomes with two telomeres at terminal ends, and all the centromere, and then URA, then TRP1, then SUP, all the components you can see. So you can see here how it has been created. So this was in the okay in the plasmid form. This was connected. So after digestion with BAM H, okay, BAM H, this get digested and it is released. It is released, and BAM is isolated from the Bacillus amniofaciens, and it has got the sequence 5 prime GGATCC. So these palindromic sequences, it belongs to, of course, the restriction enzyme 2 type. So within the palindrome sequence, it is cut in between the 2G. So this 2G is there, it is cut. So after that, what will happen? The linear chromosomes will be created, and these chromosomes can be then introduced into the yeast cells, where it can be remain stable. It can also propagate by virtue of the this ARS sequences. So the ARS, it is autonomous replicating sequences from the yeast cell, and this ARS, it has got the sequence ATT. It has got the sequence 5 prime ATTTAYRTTTT, where it is a pyrimidine, it is a purine. Any purine or pyrimidine can be there. And this ARS, it has got the four regions B3, B2, B1, A. So these four regions are very important because the B3 is responsible for binding with the ARS binding factors. And when this factor tends to bind at these very sites, then it will initiate the melting or the breaking of the hydrogen bonds. In the hydrogen bonds, they just break and it forms a single standard fork like this, and then the DNA is synthesized. Okay, so this is the initiation site, which is called as the replicon, the melting region, and this occurs at the B2 regions. Okay, origin of recognition sequence is at the B1 site. This is called the ORC, origin of recognition sequences. Okay, uh, I told you that the artificial chromosomes of yeast, as well as the plasmid form of the yak, they bears the markers. What are the markers? Markers are URA3. This is a gene on the chromosome number 5 of Saccharomyces, that is yeast, which codes for an enzyme orotidine 5' phosphate decarboxylase, ODCase, which is an enzyme that catalyzes one reaction in the synthesis of uracil. It is used to identify the chlorine in the yeast medium. Okay. Similarly, TRP1, this is an enzyme, phosphoribosyl anthranilate isomerase, an enzyme which can lead to the formation of the tryptophan. So when this URA 3 minus and TRP minus, so these are the oxytroph. They cannot synthesize in the absence of uracil. This can also cannot synthesize or form the colony in the absence of the tryptophan. So these two are the markers. And besides, one another marker is there, and that is called as the sub4. That is called as the sub4, which I will tell you later on. So here you can see that if the auxotroph will be there, if auxotroph is there, for example, say, this yeast cells, it is TRP1 minus and URA3 minus. It is URA3 minus. Okay, so this when it will allow to grow in the minimal medium, it cannot grow. This cannot form colonies because it lacks the tryptophan and the uracil, which is essential for the growth. But once the minimal medium in the minimal medium, uracil is added, or tryptophan is added, tryptophan, then such type of thing that tends to grow. Or if it is URA3 plus plus, URA plus plus, then it also grows. So these are the autotrophs. Autotrophs cannot grow in the medium, minimal medium, while the prototrophs grow in the culture medium. For example, the PDA. So, so how the DNA gets inserted into the linear vector? So for that very purpose, this has been opted from the genomic DNA. This is a genomic DNA. So with the help of the SNA1, SNA-B1, this is the enzymes restriction enzymes. So this restriction enzymes, it has been isolated from the Spherotilus natans. So when this enzyme is allowed to digest the this linear chromosomes, then it will cut at the specific site of this SNAB1, and due to this there will be a gap, and you can see that the 2 megabase means 10 to the power 6. Okay, so 2 into 10 to the power 6. You can see that what enormous size of this DNA is there. So this DNA is get inserted at this very site, and it forms a structures like this. So you can compare telomere, then you can the selective marker, you have got the ori bacteria, then sen is there, then ori site is there, then the DNA insert is there, then again the this selective marker, then telomere. So this becomes the linear chromosome. Now it is ready to get introduced into the yeast cells because it is linear. So we have got the two option. One option is that either this linear chromosomes, they have got the insert, or linear chromosome they don't have the inserts. So this is the insert is here and here. When the insert will be there, it will be called a transformant, and when the insert is not there, it is called as the non-transformant. So let us just proceed whether how we can screen out the transformants and the non-transformants. So for that very purpose, one marker is there that is called the sub4 screening marker. This is called the screening marker. Okay, so the screening marker is sub4, which encodes a tRNA. It encodes a tRNA that suppresses the ADE2 genes, that suppresses the ADE2 genes. And what is ADE2 genes? ADE2 genes 1 and 2 are the enzymes involved in the synthesis of adenine. Okay, adenine. So if sub 4 is active, or it is the wild type, then the red pigment is getting accumulated, and so whatever the colonies that will grow, they will having the red colors. Okay, and if the insert is there. If the insert is there, it means that the sub 4 is not able to, and not able to retain the red pigment. Okay, so that colony will be white. So here sub 4 plus, it has formed the red colony, and sub minus, it will form the white colony. Or better you can say the red colonies contain recombinant vectors, contains sorry, red colonies are non-recombinant, and white colonies are the recombinant yak vector with DNA. So these are the we can also better explain in this one. So here it is the integration. You can say here, uh, this is the sub 4 genes. This is the sub 4 genes where multiple cloning sites are there. So after the addition with SNAV1, a gap will be there, and this insert, this insert is getting integrated at this very site. So when it is introduced by means of the electroporations, so first you have to treat it with the some of the chemical agent which will just break the cell wall, and when the cell wall is broken, it is called as spheroplast. Spheroplast. So, okay, so this is a spheroplast in which by electroporations this a clone DNA or the vector was introduced. When this vector was introduced, it also has got the URA3, or that is TRP1. So these were the plus sign. But this yeast was mutant. So when there was no when there was you can say when there was this is the TRP minus, and this is URA3 minus. Okay, so they in the this one in minimal medium they won't form the colonies, but if the vector get inside, if the vector get inside, say, then it bears the URA plus and also TRP plus. So it will compensate the minimal medium because it will synthesize the enzymes for the uracil biosynthesis, tryptophan biosynthesis, and the colony will tend to grow. So, but the what is the basic difference in between these two is that here it is the vector without any insert. Here is the vector that is yak with inserts. So this is sub 4 genes, which is a suppressor genes. They are not inactivated; they are active. So when they are active, it will form the red colony. But here, due to insertion of this DNA, this becomes the inactive. So sub 4 becomes inactive, and when it becomes inactive, then it is not able to retain the red color, and the colony will become white. So in a nutshell, in a combined forms, those colonies which are white in color, they are said to be as the transformant, and those color which are red, they are said to be as the non-transformant. So what is the utility of this very one? This TRP1 and UR3 minus strain is used to differentiate, say, whether the yeast is URA3 minus or URA3 plus. So what will happen if it will if it will without any vector, if it will be vector without inserts, and if it will be having the vector with inserts. So these are the three options. So those without vectors, it cannot grow. It cannot grow. In this vector, it will grow and will form red colony, and this vector will grow. It will form white colony. No growth, red colony, white colony. So you can differentiate the three types of yeast growing in the minimal medium. So these are some of the advantages that it provide the largest insert capacity, capacities. Also, it is advantage over the bacterial artificial chromosomes. It can be also used as the express eukaryotic vectors for the translation of the proteins, and also for the post-translation modifications. Also, um, mm, also okay, this can be also converted into the expression vector sum. So what are the applications? Yak can be utilized to clone and assemble the entire genome of an organism. Okay, so if you want to clone, suppose this is the genomic DNA and it's large size, these very sizes are there. So in order to make the genomic library of this very, so it is better that you go for the yak vector rather choosing plasmid or cosmid, or you can say phase, lambda phase. So over all these vectors, yak is the dominant one, or it has got the more advantage. Also, it is used to find genes, segment of DNA that codes for specific proteins or phenotypes. But beside this, it has got the limitations. And this limitation is that when the very large insert is there, then that chromosome becomes very fragile and it is prone to breakage, leading to the problem of rearrangement. So if the DNA of this large size, they just break into this, that this is the A, this is the B, this is the C, D. And if rearrangement occurs, it may be the A, then C, then D, then B. So this is the mutated one. This cannot be a correct cloning. So it has the problem of breakage and rearrangement into different forms. But another limit is that it's difficult to separate yak from the other host chromosomes because of their small size. Okay, if the bacteria which are smaller than the yeast, so it is very difficult to isolate it. Or also the yield of DNA is not high when the yak is isolated from the yeast cell. So there are the some of the limitations. Hope everything has been explained. In case if queries are there, then very you are welcome to ask any questions. So thanks you very much.